Dear students, today we will talk about histology of the organs of immune system. So, immune system, it's a system which provides immunity. Immunity is the main function of immune system. What is immunity? Immunity is protection of our organism, of our inner environment from different harmful factors of outer environment. And which factors do provide this protection? We have different borderline tissues, different borders, which form border between inner and outer environment. For example, here we can see epithelium. Also, it may be covered with mucus. So, different mucous membranes, skin, they protect our inner environment from different factors. But also, we have specialized cells which do provide these immune reactions, which protect our body, our organs from different harmful factors. So immune cells or immunocytes, they are main cells in the organs of immune system. So which immune cells do we know? Which immune cells do we have? You know that there are cells of blood which are called leukocytes or white blood cells. We have two types of them, agranulocytes and granulocytes, and there are different kinds of them. There are monocytes, T lymphocytes, B lymphocytes, neutrophils, basophils, and eosinophils. And those cells, they live in bloodstream during some hours, sometimes some days. And after that, they migrate into the tissues, mainly in connective tissue, but also in other tissues, and there they provide their functions. And in the connective tissue, those cells are known as transient cells of connective tissue. So monocytes, they migrate into the tissues and they become to be macrophages. T lymphocytes, they become to be tissue lymphocytes. B lymphocytes become to be plasma cells. Neutrophils in connective tissue are macrophages. Basophils become to be mast cells, or they are related with mast cells, and eosinophils, they migrate into the tissues and they are tissue eosinophils. So transient cells of connective tissue and some other tissues, they provide immune reactions and immune defense inside our inner environment. And here we can see leukocytes in blood. Here we can see lymphocytes, monocyte, neutrophils, eosinophil, basophil, and they migrate into the tissue. And here we can see a histological picture which shows us loose fibrous connective tissue. And here we can find macrophage, mast cell, and lymphocytes and plasma cell. So they provide immune reactions inside connective tissue. But also we have organs which are specialized in immune reactions, and their basis is composed of reticular tissue. Here we can see reticular tissue and it's composed of reticular cells. Here we can see them. Reticular cells and reticular tissue, they form network. Rete, it means network. That's why this tissue is called reticular. And this network provides support for immune cells. So here we can see reticular tissue. There are reticular cells. They have star-like branched shape, and also they have bases of reticular fibers. They maintain them, and they also form three-dimensional network. And here we can see empty reticular tissue. It's known as reticular stroma of immune organs. It's present in almost all immune organs except thymus, and we will see which tissue is present in the thymus. So organs of immune system, they have reticular stroma. It's a supportive part of the organ. It doesn't provide immune defense, but it forms microenvironment for the developing immune cells. And now we can see immune cells which fill the spaces between reticular cells. Here we can see them round, mainly lymphocytes are present here, and they are main cells in the immune organs, so they provide immune functions, they provide immune defense, and reticular stroma, this stroma, it forms microenvironment for the developing and resting and functioning immune cells. So here we can see two main components of the immune organs. Reticular stroma, it's composed of reticular tissue and immune cells, different immunocytes, which undergo development, differentiation, and they are functioning here. And also immune organs, they are like home for immune cells, and they migrate into the connective tissue, they provide immune reactions, and then they come back to immune organs where they are resting, they undergo differentiation and in other reactions. So it's like home for immune cells, and coming back to immune organs is called homing. It's process of migration of immune cells to the immune organs. So there are some differences between connective tissue. Here we can see connective tissue with immune cells. They migrate here and they directly provide immune reactions. And in the reticular tissue, in the immune organs, hemopoietic organs, reticular tissue forms stroma, it forms microenvironment, and those cells migrate here and they fill the spaces between reticular cells. And here we can see a histological picture of reticular tissue. Here we can see reticular cells, branched cells, and between them we can see leukocytes developing, leukocytes or resting leukocytes. And here we can see resting stroma, empty stroma without leukocytes, and stroma filled with leukocytes. And usually under microscope we cannot distinguish all those violet points, and those points mainly are nuclei of leukocytes, of lymphocytes, immune cells, but also between them we can find reticular cells, which also play important role in the functioning of immune organs. So what immune organs do we have? We have such immune organs as thymus, red bone marrow, spleen, lymph nodes, tonsils, and also mucosa-associated lymphoid tissue, which in some classifications includes tonsils. And all those organs may be divided into two main groups. There are immune organs which are called central and peripheral immune organs. Here we can see central and peripheral immune organs. Central organs include red bone marrow and thymus. Here we can see red bone marrow and thymus. And all others are peripheral organs. They include spleen, lymph nodes, tonsils, and mucosa-associated lymphoid tissue. So what is the difference between central and peripheral organs? Both groups of organs provide differentiation of different immune cells. But this differentiation may be antigen independent and dependent. Antigen independent differentiation is differentiation of naive cells when they didn't participate in the immune reactions. And immune cells, when they have contact with antigens, they undergo antigen-dependent immune differentiation, and it takes place in the peripheral organs. So what is antigen? Antigen is very specific part of the proteins, of another substances which are located in the substances in different particles, for example in bacteria, and antigens are recognized by immune system. It's a like face of different bacteria, viruses, and immune system recognizes them using antigens. So when cells meet antigens, they undergo antigen-dependent differentiation, and it takes place in the peripheral organs. But before this, immune cells undergo antigen-independent differentiation, and they are developed in central organs. So red bone marrow, it provides hemopoiesis, formation of blood cells including leukocytes, and its formation of leukocytes. And some of them migrate into the thymus, there are T lymphocytes, and in those places they undergo antigen-independent differentiation without contact with antigen. So antigens are absent there. And only after that, after first stage of their development, they migrate in the peripheral organs where they undergo antigen-dependent differentiation and they provide their functions. And also after second stage of their development in the peripheral organs, they migrate into the tissues, into the organs. Where do they provide their functions? They provide immune defense, immune reactions. For example, they participate in the inflammation, and they may come back into the peripheral organs, and we call this homing. So first stage, it's development of cells and antigen-independent differentiation in the central organs. Second stage is antigen-dependent differentiation in the peripheral organs, and third stage it's participation in immune reactions in connective tissue and in different organs and tissues. It's third stage of life cycle of immune cells. And now let's look at histological structure of central and peripheral immune organs. And we'll begin from peripheral immune organs. And here we can see a histological slide which shows us tonsil, its lymph node and spleen. And we can find here that those violet points, those violet round structures are lymphocytes, different numerous lymphocytes, immune cells. And we can see here that immune cells, lymphocytes, they form spheric structures, round structures, they form groups. And here we can see this group of lymphocytes. So how do we call this group? This group of lymphocytes is called lymphatic nodule or lymphatic follicle. Don't confuse two terms, lymph node, its organ, and lymphatic nodule or lymph nodule, it's small group of lymphocytes. This is nodule and this is node. Or nodule may be called follicle, lymphatic follicle or lymphoid follicle. So lymphoid or lymphatic follicles or nodules, they are small groups of lymphocytes which are present in tonsils, in the lymphatic nodes, in spleen, and also they may be present in mucosa and submucosa of different hollow organs. For example, in digestive tube, in respiratory system, and lymph node, it's organ of immune system. So don't confuse two terms, lymph node and lymphatic nodule. Nodule is small group of lymphocytes which are present in different peripheral organs, and lymph node, it's organ. And here we can see lymphatic nodule or lymphatic follicle, and there are numerous lymphocytes. They are present here, and we have light center, it's germinative center of lymphatic follicle or lymphatic nodule, and its mental zone or peripheral zone, it's darker than central zone. Central zone, it's a zone of development of leukocytes. They undergo divisions here, so it's called germinative center. And peripheral zone, it contains mainly resting or differentiated lymphocytes. So let's begin from mucosa-associated lymphoid tissue. It has the simplest structure. Here we can see mucosa of gastrointestinal tract, and in the loose connective tissue which underlies epithelium, we have groups of lymphocytes which provide immune defense. So they are located directly under epithelium in mucosa or in submucosa, and there they provide immune defense. And we eat a lot of harmful substances, we eat a lot of bacteria, and they may cause different diseases. And we have lymphocytes in connective tissue of our mucosa, and those lymphocytes, they provide immune defense. So here we can see lymphatic nodules. Here we can see, or lymphatic follicles, they are called groups of lymphocytes which provide immune defense. Here we can see lumen of the organ, and different bacteria are present here, but lymphocytes, they protect underlying wall from different harmful factors. It's mucosa-associated lymphoid tissue, and it includes gut-associated lymphoid tissue, which is present in gastrointestinal tract, or it's called GALT, bronchus-associated lymphoid tissue, which is present in respiratory system, and also it includes SALT, or skin-associated lymphoid tissue, which is located in the connective tissue of skin. So MALT or mucosa-associated lymphoid tissue includes lymphoid tissue of gastrointestinal tract, of respiratory system, and lymphocytes of skin. And also we have tonsils. In some classifications, tonsils are described as part of mucosa-associated lymphoid tissue. So what is tonsil? Tonsil, it's groups of lymphocytes which are present in mucosa of oral cavity, pharynx or larynx according to the localization of the tonsils. And some tonsils are located in the oral cavity, palatal tonsils, lingual tonsil, they are present here. Also, some tonsils are present in pharynx and in the larynx, and they are formed by mucosa of those organs. And here we can see mucosal folds. They form crypts. They are invaginations of mucosa. And near the crypts we have groups of lymphatic nodules. Here we can see them. They surround crypts. And when something enters in our oral cavity, for example, some bacteria, they may enter in those crypts, and in the crypts, near the crypts, we have numerous lymphatic nodules which do provide immune defense here. So it's shape of the tonsil, and covering of the tonsils depends on localization. Those tonsils which are located in the oral cavity, they have stratified squamous non-keratinized epithelium, the same as in the oral cavity. And tonsils of the larynx, pharynx, according to their localization, they have pseudostratified epithelium, ciliated pseudostratified epithelium, which corresponds to those mucosa of those organs. So here we can see epithelium. Under the epithelium we have loose fibrous connective tissue, and it's surrounded with numerous follicles. And here we can see single crypt. We can see here a crypt. It's a drawing of histological site, and it's histological microphotograph. So we can see here a crypt. Epithelium corresponds to localization of this tonsil. So we have here palatal tonsil, and epithelium is stratified squamous non-keratinized. And there are numerous lymphatic nodules or follicles which surround this crypt. And some lymphocytes, they excite from the lymphatic nodules, and they migrate between the cells of the covering epithelium in the crypt. Here we can see lymphocytes inside the epithelium, and they may enter in the cavity of the crypt. It's invagination of epithelium, and they may come back to the lymphatic follicles or lymphatic nodules. It's structure of the tonsil. Palatal tonsil and another tonsil, they have a lot of similarities in their structure, but main difference is different kind of epithelium. This kind of epithelium corresponds to epithelium of mucosa of those organ where this tonsil is located. And now let's go to lymphatic nodes. Don't confuse lymphatic nodule, small group of lymphocytes, and lymphatic nodes. They are organs, peripheral organs of immune system. And main function of lymphatic nodes is filtration of lymph. So let's look where do we have lymph and how lymph is developed. Here you can see microcirculation. There are blood vessels, arteriole, capillaries, and venule, and they provide circulation in the connective tissue, for example. And here we can see lymphatic capillary. It has a blind end. And water, some amount of fluid, comes here via arterial vessels, via arterioles, and it comes to the capillaries, where it penetrates wall of the capillary and goes to the tissue. So from the capillaries, water goes to the connective tissue, to the intercellular substance of connective tissue. And after that, some amount of water is uptaken by blood vessels. It goes to the capillaries and venules. But some amount of water also goes to the lymph, to the plasma of lymph. It's filtrated and it goes to lymph. It's a place of development of plasma of lymph. So it's how lymph is developed. Lymph contains plasma, fluid, intercellular substance, and also it contains formed elements of lymph, and formed elements of lymph are leukocytes. 98-97% of them are lymphocytes. And they are called lymphocytes because they are predominant type of formed elements of lymph. So lymph uptakes some amount of intercellular substance of connective tissue, and now it's called plasma of lymph. And some lymphocytes may migrate from the connective tissue, from the place of immune reaction, to the lymphatic capillary, and they are present in this lymph. This lymph is primary lymph, which is a result of filtration of tissue fluid, and it may be dirty. It means that it may contain different harmful substances, it may contain different bacteria, viruses, etc. And it cannot go directly to the bloodstream because it may cause different diseases. And this lymph should undergo filtration, and this filtration takes place in the lymphatic nodes. So here we can see blood circulation, arterial part, capillaries and venous part, but also some amount of fluid goes to the lymphatic capillaries. They provide filtration, and then lymph goes to the efferent lymphatic vessels, big lymphatic vessels, which are similar to veins, but they contain lymph. And they bring this primary lymph to the lymphatic node. And lymphatic nodes, they provide filtration. They also are a place of differentiation of lymphocytes, and after this filtration, lymph goes to the efferent lymphatic vessels. Also, some lymphocytes may excite from lymph node and go to the efferent lymphatic vessel. And these vessels, they bring lymph to thoracic duct, which is opened into veins. And here lymph and venous blood are mixed. So lymphatic nodes, they provide filtration of tissue fluid, which is called lymph in lymphatic vessels. And here we can see a structure of lymphatic node. It has bean-like shape. It has a portal part. It has connective tissue capsule, which covers lymphatic node. There are efferent lymphatic vessels and efferent lymphatic vessels. Efferent, they bring lymph to the lymphatic node, and efferent, they carry lymph away from lymphatic node. So on this part, near the capsule, we can see efferent lymphatic vessels which carry lymph to lymphatic node, and here in portal part we can find efferent lymphatic vessels. And also in the portal part we can find artery. It enters via this region to the lymphatic node, and vein, it excites from lymphatic node. So lymphatic nodes, they also have blood supply. And also there are nerves which also enter via portal part of the lymphatic node. So lymphatic node has portal part, it's beginning of lymphatic vessels, and also there are blood vessels here and nerves, and it has connective tissue capsule, and under the capsule we have openings of the efferent lymphatic vessels. Connective tissue capsule has processes which are called septa. They penetrate peripheral part of the lymphatic node. And here inside the lymphatic node we can see numerous lymphocytes which form lymphatic nodules. Here we can see them, and also some structures which aren't like lymphatic nodules. So lymphatic node, it has three regions according to the structure. Its peripheral zone, it's called cortex. Cortex is composed of lymphatic nodules. Here we can see lymphatic nodules, and this region is called cortex. It's outermost part of the lymphatic node. The innermost part of the lymphatic node, it's medulla. Here we can see medulla, and it's composed of medullary cords. They aren't nodules, they are elongated and branched groups of lymphocytes. We can see them here. And between medulla and cortex, intermediate region is called paracortical zone. Here we can find paracortical zone. So all three zones, cortex, paracortical zone, and medullary cords, they contain reticular tissue, which is basis of lymphatic node. And in the reticular tissue, we can see numerous lymphocytes, and we cannot distinguish where is reticular tissue, where are lymphocytes, but all these points are nuclei of reticular cells and mainly nuclei of lymphocytes. So lymphatic nodules, paracortical zone, and medullary cords of medulla of lymphatic node. So here we have cortex. It's composed of lymphatic nodules or lymph nodules, medullary cords which form medulla, and paracortical zone, diffuse lymphoid tissue without any structural different structures like lymphatic nodules or medullary cords. It's only diffuse lymphoid tissue. And we can see that on this diagram, cortex, medulla, and paracortical zone, they have different shades. It's because of distribution of different kinds of lymphocytes. If you remember, there are two kinds of lymphocytes, T and B. And T and B lymphocytes, they have different distribution in the immune organs. So the more numerous lymphocytes here are B lymphocytes, and they have two zones of their localization. First zone it's cortex, it's B dependent zone, and second zone it's medulla, also B dependent zone. And paracortical zone it's T dependent zone, and T lymphocytes are present here. So B, T, and B zone. Cortex B dependent zone, paracortical zone T dependent zone, and medulla it's B dependent zone. Different kinds of lymphocytes are distributed in different parts of lymph nodes. And here we can see that basis of lymph node is reticular tissue, and between reticular cells we can find numerous lymphocytes. And let's look at a diagram which shows us microscopic structure of lymph node. Here we can see connective tissue capsule, and here we can see gap between capsule and tissue of the node. This gap is called lymph sinus. It's filled with lymph. And it's like blood vessel, but it's not blood vessel, it's gaps, lymph sinuses inside the lymph node. There are gaps between groups of lymphocytes, between lymphatic follicles. And here we can see efferent lymph vessel. It also has inner lining with endothelial cells. So lymph comes here and goes in the gaps inside the lymph node. Here we can see reticular tissue. It forms stroma basis network which will support, which will maintain lymphocytes. And also reticular tissue is present in the gaps in the lymph sinuses. So not only in the lymphatic nodules, in the medullary cords we have reticular tissue. This tissue also fills all gaps inside the lymphatic node. So now we can see a stroma of lymphatic node and its network. It's like sponge-like structure. And also this stroma contains macrophages which develop from monocytes of blood. Here we can see macrophages. And also there are lymphocytes, numerous lymphocytes. In this case we have cortex, and there are B lymphocytes. They fill the space between reticular cells. So B lymphocytes, they fill the spaces between reticular cells. They form lymphatic nodules here, and between nodules and capsule we have gap. It's sinus of the lymphatic node. And its lymphatic vessel, efferent lymphatic vessel, it carries lymph here. And between lymphatic follicles of the cortex we have cortical sinus. So this gap also is present here. And why do we have those gaps? They are ways where lymph goes through lymphatic node, and they provide filtration of lymph inside lymphatic node. So let's look at this diagram and this lymphatic node. Here we have capsule and efferent lymphatic vessels. Here we can see also capsule, it's this region, and lymphatic vessel. It carries lymph here. And there are lymphatic nodules. You can see them here. And we can find here white regions. They are gaps. They haven't this dense tissue, lymphoid tissue, they are filled only with network of reticular cells. They are called sinuses of the lymph node. And we have here subcapsular sinus. It's located under the capsule, or peripheral or marginal sinus, subcapsular sinus. Then between the lymphatic follicles we have cortical sinuses. Then lymph goes to the medullary sinuses, which are present between medullary cords, and then lymph goes to the portal sinus, and from portal sinus it goes to the efferent lymph vessels. So it's way of filtration of lymph in the lymph node. Lymph comes here via efferent lymph vessels. It's distributed under the capsule inside the capsular sinus or marginal sinus, and it goes in the cortical sinus, then in medullary sinus, and goes to the portal sinus, which is the beginning of the efferent lymph vessels, which carry lymph away from the lymph node. And all those sinuses, they have this network of reticular tissue which helps to filtrate lymph. So we can see here this structure, and let's look how is it working. On this picture, on this diagram, we can see subcapsular sinus and cortical sinus, and it's followed by medullary sinus and portal sinus. They are located deeper. So let's look. Here we can see cortical part, its cortex, and efferent lymph vessels. They carry different cells, they carry different harmful substances, and this lymph node, it filtrates all those substances, and then only useful substances excite from the lymph node. Also useful cells, immune cells, they excite from the lymph node and go to the bloodstream via efferent lymph vessels. So here we can see capsule with efferent lymph vessel, endothelium which lines sinuses inside the lymph node, reticular tissue of lymphoid tissue of the cortex, also reticular tissue which fills the spaces in the lymph sinuses. Macrophages are present here, and lymphocytes, numerous lymphocytes are present here. And now let's look. It's lymph, it contains some lymphocytes, and lymph is distributed under the cortical sinus. Some amount of lymph comes up, comes down, and some amount of lymph goes between lymphatic follicles via cortical sinuses. So here we can see distribution of lymph and flow of lymph inside the lymph node. And for example, something harmful comes here with dirty lymph, which is a result of filtration. For example, bacteria. And here we can see that bacteria meets macrophage, and macrophage it eats this bacteria. It provides phagocytosis, so it neutralizes this bacteria, and it helps to provide immune reaction. And here we can see another bacteria. It goes to another macrophage. It also uptakes this bacteria and provides immune defense. So it's how lymph node is working. It provides filtration of lymph from different harmful substances. And after this phagocytosis, macrophages, they will conduct information about antigens of those bacteria to lymphocytes, which are developing here. You remember that they undergo antigen dependent differentiation here, and they will receive information about those antigens, and activated lymphocytes will excite from the lymph node, and they will go to the efferent lymph vessels and to the bloodstream, where they will participate in the immune reactions. So here we can see release of lymphocytes, activated lymphocytes to the lymph, and then they will participate in the immune reactions. And in some regions, mainly in the medulla, we have plasma cells, and for example, we will show plasma cell here. And plasma cells, they produce antibodies. They develop from B lymphocytes, and they produce antibodies. And here we can see antibodies, they have Y-like shape, and they are released in bloodstream from the plasma cells. So plasma cells, after receiving information about some antigen, it produces numerous antibodies which provide humoral immune reactions. It's also very important for the function of immunity. So it's how lymph node is working. It provides filtration, it provides differentiation of lymphocytes, and also production of antibodies by plasma cells, which also are present here. So lymph nodes, they filtrate lymph, and also they provide some immune reactions, and we can see the structure of the lymph node. And now we can see that dirty lymph goes from the tissues, undergoes filtration in the lymph node, goes to the efferent lymphatic vessel, and goes to thoracic duct where lymph is mixed with venous blood. And here we can see histological slide which shows us microscopic structure of the lymph node. Here we can see lymph node, its cortex, paracortical zone. In the cortex we can find lymphatic follicles or nodules, and medulla of the lymph node with medullary cords. They are denser, and between them we can find sinuses of the lymphatic node. There are nodules, its subcapsullary sinus, and its paracortical zone, and deeper we have medulla with medullary cords and medullary sinuses. And now we are going to spleen. It's also organ, peripheral organ of immune system. And here we can see structure of spleen. Spleen also has capsule, connective tissue capsule, and also it has septa or trabecular processes of the capsule which penetrate tissue of the spleen. They enter inside the spleen. Spleen is covered with peritoneum. It has mesothelium, simple squamous epithelium, which covers the spleen, and connective tissue of peritoneum is fused with connective tissue of capsule of spleen. So we can see here mesothelium, simple squamous epithelium. Under this we can see a layer of connective tissue of capsule of spleen, and also septa or trabecula of spleen which penetrate tissue of spleen. And specific feature of connective tissue capsule and septa or trabecula of spleen is that connective tissue contains bundles of smooth myocytes. They are present here, and that's why spleen is able for contraction. Connective tissue capsule of spleen and trabecula, which maintain the shape of the spleen and protect the spleen, they may be contracted. Why do we need this? This contraction pushes out different formed elements which are present in the tissue of spleen, and it's needed, for example, in the hemorrhages. When your patient will may lost a lot of blood, his spleen will be contracted and will push out some amount of formed elements of blood, for example erythrocytes, and they will help to survive in severe hemorrhages, for example. It's protective and supportive part of spleen, capsule, and trabecula, which have ability for contraction. And inside the spleen we can see tissue of spleen, different structures, numerous points, red points, violet points, and all this tissue of spleen is called pulp. Here we can see pulp of spleen. And pulp of spleen has two kinds, or it may be divided into two kinds. It's red pulp and white pulp. Here we can see lymphatic nodules, violet round structures. They belong to white pulp. So white pulp on this picture has violet staining. And between white pulp we can see regions filled with something red, with something violet. It's called red pulp of spleen. Here we can see red pulp and white pulp. And on the section of spleen, in the anatomical preparation without any staining, we can see white regions with whitish shades. And there are lymphatic nodules. They contain lymphocytes. Lymphocytes are white blood cells, and they have white shades. And we can see them on the section of the spleen. It's white pulp. And red regions, which contain mainly erythrocytes, are called red pulp. That's why in unstained preparations, white pulp has light shades, whitish shades, and red pulp it's dark red. And in histological sites, in hematoxylin and eosin staining, white pulp is violet and red pulp is red or pink. So white pulp it's part of spleen which includes mainly lymphatic nodules, but also it includes periarterial lymphoid vagina, which surrounds arteries inside the spleen. So cylindric structures which are following arteries inside the spleen. And also there are lymphatic nodules. And here we can see a special type of blood supply of spleen. There are trabecular and spleen arteries. They are divided into branches which enter in the trabecula of spleen. So those arteries inside the trabecula are called trabecular arteries. They are located in the trabecula. And then they excite from trabecula, their branches excite from trabecular regions, and they are called central arteries. It's central artery of spleen. It goes into pulp. And trabecular artery, it's bigger and it's located in trabecula. So it's pulp artery, which is called central artery. And it penetrates lymphatic follicles or lymphatic nodules, so it's located inside the lymphatic nodules. That's why a specific feature of lymphatic nodules of spleen is presence of artery. They are only lymphatic nodules which may contain artery. If you see lymphatic nodule with blood vessel inside, you can conclude that it's a histological site of spleen. And lymphatic nodes or mucosa-associated lymphoid tissue, tonsils, they don't have artery inside the lymphatic follicles. So it's specific feature of spleen. And central artery penetrates these follicles and excites from the follicles and is divided into short branches which are called penicillar arteries. And they are opened or in the splenic sinuses, some kind of the capillary, or they are freely opened in the pulp of spleen, and we will see how it is working. So here we can see lymphatic follicle of spleen. It surrounds central artery. It's white pulp of spleen, and you should know structure of lymphatic follicles of spleen. Here we can see first specific feature of it, its presence of artery. It's present inside the lymphatic follicle, and it's called central artery, but it has eccentric localization. It's not localized inside the center, exactly in the center. It's located near the center. So first of all, lymphatic follicle contains central artery. Near the central artery we have special zone which is called periarterial zone of lymphocytes. This zone surrounds artery of the lymphatic follicle. Near this follicle we can see light center which is called germinal center. Here we can see it, and you should know that periarterial zone is T-dependent zone and germinal center is completely B-dependent zone. This zone is T-dependent, this zone is B-dependent zone. Here we can see T and B-dependent zones. And also we have mental zone which surrounds both. Here we can see two zones previous, and they are surrounded with mental zone. This mental zone is mixed T and B at the same time, but B lymphocytes they prevail. So mainly B lymphocytes are present here. And also we have fourth zone in the lymph follicles of spleen. It's marginal zone. Peripheral zone, the most peripheral zone is marginal zone. So we have four zones of lymphatic nodules in spleen: periarterial, germinal center, mental zone, and marginal zone. T-dependent zone, it's periarterial zone. B-dependent zone, completely B-dependent zone, it's germinal center. Mental zone as well as marginal zone, they are T and B dependent zones at the same time, but B lymphocytes, they prevail here. So mental zone and marginal zone, they are mixed, and completely T dependent zone is periarterial zone, and completely B dependent zone, it's germinal center. So please remember 4 regions of lymphatic follicles of spleen: periarterial zone, germinal center, mental, and marginal zone. And here we can see lymphatic nodules or lymphatic follicles of spleen. We can see central artery. Near this central artery we have periarterial zone, is germinal or light center, mental zone, and marginal zone. And here we can see in other regions, here we can see capsule, trabecular artery, and here we can see central artery, and all other structures. Here we can see diffuse tissue with red cells, violet cells. It's called red pulp. It has little bit different structure, different from the white pulp. So white pulp, it contains mainly lymphatic follicles, and red pulp, it contains splenic cords, which are alike medullary cords of lymphatic nodes, and between them there are splenic sinuses, which are alike medullary sinuses of lymph nodes, but splenic sinuses are filled with blood, not with lymph. And let's look how is it working. So here you can see reticular tissue, which also is basis of spleen. It's also reticular stroma of spleen, which is similar to reticular stroma of lymphatic node. Reticular cells and reticular tissue. And here we can see endothelium, which forms wall of the splenic sinuses. There are medullary sinuses in the lymph node, and almost the same are present in the spleen, and they have endothelial lining. So we can see here, and you remember that central artery opens in the penicillary arterioles, which may freely be opened in the tissue of spleen pulp, or they are continued by splenic sinuses. They are modified capillaries. They are very wide in diameter, and they have gaps inside their wall, and so they are very wide capillaries, and they have specific structure, specific feature inside the spleen. And also we have macrophages here, as well as in the lymph nodes, and numerous lymphocytes, and also there are numerous erythrocytes here. That's why the red pulp is called red. So here we can see tissue of medullary cords. They have irregular shape, different shape, polymorphic shape, and between them we have cavities filled with blood. Those cavities are splenic sinuses, which are modified capillaries of sinusoidal type. And here we can see medullary cords. They are darker, and between them there are splenic sinuses filled with blood. Here we can see splenic sinus filled with blood. So there are 2 types of blood supply of spleen. First of all, trabecular artery gives branches, which are central arteries, which are going through the pulp, through white pulp, and then they excite from white pulp. They are not surrounded here with lymphatic follicles. They excite from them, and they are divided into the penicillar arterioles or penicillar arteries. Some of them are continued by splenic sinuses, very wide capillaries, and some of them have complete direct opening into the pulp of spleen. And here we can see trabecular artery, and its central artery. It penetrates central part of the lymphatic follicle of spleen, or little bit eccentric part. Its periarterial zone, its germinal center, its mental and marginal zone, and this artery excites from the follicle, and then it may be continued by sinuses. It enters in the sinuses, very wide capillaries, or it's freely in this place, blood freely goes to the blood of spleen. That's why we have 2 types of blood supply of spleen. It's closed when blood doesn't excite from bloodstream, and opened when blood freely goes to the red pulp of spleen. Two types of blood supply. And let's look how is it working. Blood comes here, and in closed type of blood supply it goes to the sinuses and doesn't left bloodstream. And in some cases some amount of blood goes freely to the pulp of spleen, and it's like spongy-like structure, and blood goes freely in the red pulp. So two types of blood supply of spleen. And also arterioles of spleen, they have sphincters which regulate the blood flow, and they regulate functioning of two types of blood supply of spleen. So some blood goes to sinuses directly, some blood goes directly to the red pulp of spleen. And then also spleen participates in the destruction of old erythrocytes. Let's look. Leukocytes, lymphocytes, for example, they excite from tissue via gaps in the endothelium. Look here. They are released in bloodstream, and also some erythrocytes, they go to the red pulp and they are distributed in the red pulp, and then they excite via gaps in the endothelium, they excite to the bloodstream. But old erythrocytes, they become to be spheric in shape. They are spherocytes. And normal erythrocytes, they have biconcave shape. But old erythrocytes, they are spheric and they cannot pass through these gaps in the endothelium of spleen. And here we have macrophages which eat them. So here we can see normal erythrocyte and old spheric erythrocyte. Here we can see migration of normal young erythrocytes between gaps in the endothelium of splenic sinus. Here we can see splenic sinus, and there are endothelial cells. They have specific shape, they are elongated, and in the gaps between them there are gaps via which erythrocytes may migrate to the red pulp, and from the red pulp to the bloodstream. And these spheric erythrocytes, they cannot pass through this gap, and here we have macrophages which uptake them, and they provide phagocytosis of old erythrocytes. That's why one of the functions of spleen is breakdown of old erythrocytes. And now we are going to the central organs of immune system, and we will begin from red bone marrow. So red bone marrow is organ which provides hemopoiesis, formation of all formed elements of blood, and also leukocytes. They are formed elements of blood, and they develop in the immunocytes, cells of immune system. So all cells of immune system, they begin their development in the red bone marrow. That's why this organ is central organ of immune system, because it provides formation of immune cells, and also it provides their antigen independent differentiation. Here you can see places of distribution of red bone marrow, and it's located inside the red bone marrow cavities, in the diaphysis of long bones, and also between trabecula of short bones, and in the epiphysis of short bones between trabecula of spongy bone. So all those spaces are filled with red bone marrow. Here we can see trabecula of bony tissue, and between them red bone marrow cavities are filled with red bone marrow. And here we can see trabecula. There are osteoblasts which cover this trabecula, osteocytes inside trabecula, and osteoclast. It's a typical trabecula during osteogenesis. In spongy bone, we have a little bit another appearance of trabecula. We can see here also osteoblasts, but they are flattened. They aren't so high because they are inactive, and also they may be called bone lining cells in this case. Also we have osteocytes inside the trabecula, and osteoblasts or bone lining cells, they have important role because they cover bony trabecula and also they participate in the creation of microenvironment for the developing hemopoietic cells. Also, we have here trabecular reticular tissue near the trabecula of bones. So, trabecula of bones, spongy bone, it forms dense stroma, which is like a capsule in the spleen and in the lymph node, capsule and septa and trabecula. They maintain their shape. There is dense connective tissue. And in the red bone marrow, this dense stroma is formed by bony trabecula. And those septa, trabecula, capsule, and in the red bone marrow, bony trabecula, they maintain reticular tissue, which also maintains hemopoietic and immune cells. So, reticular tissue, it has own basis, it has own maintenance and support, and this support is provided by bony trabecula. And here we can see formation of bone, and there are bony trabecula of immature bone, and in the mature bone, also in spongy bone, we can see branched bony trabecula and a neighboring reticular tissue which forms reticular stroma for the red bone marrow. So let's look here. Here we can see bony trabecula of the bone, spongy bone, and here we can see reticular tissue which forms soft stroma or reticular stroma of the red bone marrow. And here we have also adipose cells or adipocytes, which also participate in the formation of microenvironment for the hemopoietic cells. Also, we have here macrophages, which provide immune functions, and also some macrophages, they are known as nurse cells, and they participate in the hemopoiesis. They regulate some kinds of hemopoiesis. And also we have here sinusoids, white capillaries, modified capillaries with white gaps in their wall. And numerous hemopoietic cells, we can see them here, and they are developing cells on different stages of their development. And here we can see the biggest hemopoietic cell, it's megakaryocyte, which is progenitor of thrombocytes or blood platelets. And here we can see formation of blood platelets. Small particles of cytoplasm of megakaryocyte are separated from it, and they are going to the bloodstream. And some leukocytes also excite from stroma of red bone marrow, and they achieve bloodstream via wall of the sinusoid. And also erythrocytes, after their development, they are released via gaps in the sinusoids. They are going to the bloodstream. So, sinusoids of the red bone marrow, they a little bit remind us of sinusoids of spleen or sinusoids of sinuses of lymph node, because they have wide gaps via which leukocytes and another cells may migrate from reticular stroma to the bloodstream. And here we can see histological slide of red bone marrow. Here we can see trabecula, adipocytes, sinusoids, and numerous and very diverse hemopoietic cells, which are progenitors of all kinds of formed elements of blood. And here we can see histological slides. We can see here different hemopoietic cells on different stages of their maturation. And here we can see bony trabecula, and all those violet points are developing hemopoietic cells on different stages. And here we can see red bone marrow, and also we have a kind of bone marrow which is called yellow bone marrow. It contains mainly adipocytes, and in adults, we have mainly in diaphysis of bones, we have mainly yellow bone marrow, because those bones which are filled with red bone marrow, they are enough to provide hemopoiesis in adulthood. And some places of red bone marrow are replaced with adipose tissue. It's yellow bone marrow. It's like empty stroma without hemopoietic cells. So it's extra place for hematopoietic cells, and for example after hemorrhages hematopoietic cells may fill yellow bone marrow and it again will be red bone marrow. And after that, after regeneration of the lost formed elements, red bone marrow may again come back to yellow bone marrow. So yellow bone marrow it forms extra places, extra stroma for hematopoietic cells, but it's not functioning, it's not working at this moment, but it may participate in hematopoiesis in some conditions. And here we can see red bone marrow and yellow bone marrow. It's bony trabecular and empty stroma without hematopoietic cells, and they may fill this stroma in some cases. Structural and functional unit of red bone marrow is hematopoietic islet, and basis of hematopoietic islet it's macrophage or nurse cell. It maintains hematopoietic cells on different stages, and also it forms microenvironment for them, and also it gives them different nutrients, different factors of hematopoiesis, different poietins. So it's nurse cell which provides which regulates hematopoiesis and helps in the hematopoiesis. And some broken cells, hematopoietic cells, may be eaten by this nurse cell via phagocytosis. So different damaged cells are eliminated via phagocytosis by the central macrophages of hematopoietic islets. And here we can see macrophage, central macrophage of hematopoietic islet, and numerous hematopoietic cells, family of hematopoietic cells, which surround this nurse cell. So here we can see bony trabecular, reticular stroma, adipocytes, macrophages, sinusoids, hematopoietic cells, and all those hematopoietic cells may be divided into 6 stages of development, 6 classes of hematopoiesis. There are stem cells, hemistem cells, unipotent cells, blasts, maturing cells, and mature formed elements of blood. First 4 classes, they are very similar to each other, and they are alike lymphocytes. 5th class, it includes maturing cells. It includes developing cells which undergo differentiation, and they undergo some specific features of their appearance. And only cells of 6th class, 6th stage of hematopoiesis, they may left red bone marrow and they are going to the bloodstream. And then some cells, they migrate in the peripheral organs of immune system. Leukocytes, for example, T and B leukocytes, they migrate into the peripheral organs, and there they undergo their development and antigen-dependent differentiation. But there is one exception from this system. This exception is development of T lymphocytes. T lymphocytes on the stage of 3rd class of hematopoiesis, on the 3rd stage, they left red bone marrow and they are going to the thymus, and they continue their development in the thymus. That's why they are called T lymphocytes, because they develop in the thymus, and thymus continues their development and their antigen-independent differentiation. That's why thymus also is central organ of hematopoiesis. All other cells, they complete their development in the red bone marrow. And now we are going to thymus. Here we can see localization of thymus, and let's look at the structure. Thymus, it hasn't lymphatic follicles. Here we can see that it hasn't follicles. It has a lobulated structure. Here we can see lobules of the thymus. It's covered with connective tissue capsule. This capsule has septa, which divide thymus into the lobules. Here we can see septa and lobules of thymus. Those violet polygonal structures, they are lobules of thymus. So in the thymus we haven't follicles, but we have numerous lymphocytes. They are T lymphocytes on different stages of their development, and they form lobules. So capsule, septa, which remind us of previous organs, but septa divide tissue of thymus into the lobules, and structural and functional unit of thymus is lobule. Here we can see lobule. Each lobule has two regions, its cortex, peripheral zone, and medulla, central zone. And we can see that cortex is dark and medulla is light. Medulla, central zone, and cortex, its peripheral zone. Basis of thymus, it's stroma, but this stroma, look here, this stroma isn't reticular tissue, it's a little bit different. And this thymic stroma is epithelial tissue, it's epithelial stroma. And in this case, we should say that thymus is the only organ in our organism where epithelial tissue forms stroma. So epithelial tissue usually forms parenchyma, it doesn't form stroma, it doesn't provide support for another cells. But in the thymus, epithelial tissue forms epithelial stroma for the thymus. And in the immune system, all organs, they have stroma which is composed of reticular tissue, it's reticular stroma of the immune organs. But there is one exception, it's thymus. In the thymus, epithelial tissue forms stroma of thymus. So it's main difference between thymus and all previous organs of immune system. It's epithelial tissue, that's why it has basement membrane. Basement membrane, it surrounds thymic lobule, it forms border between thymic lobule and septa. And also some regions, here we can see capsule or septa, it's connective tissue. And also basement membranes may be present inside the thymic lobules. Here we can see cells of the reticular stroma, which are of three types. The most numerous cells, they are supportive or maintaining cells, or also they may be called nurse cells. They are present here. Also, some of them have ability for the synthesis, and they are synthetic cells or endocrine cells. And third type of epithelial cells of thymus, stroma cells of thymus, it's cells of Hessal corpuscles or Hessal bodies. Here we can see inclusions which are alike rows. They are located mainly in medulla of thymus, and they contain pink flattened cells which form concentric structures like roses. And there are Hessal bodies or Hessal corpuscles. Their function is unknown, and some scientists think that Hessal bodies are composed of old nerve cells and endocrine cells of thymic stroma. So when they are becoming older, they are going to the center of the thymic lobule, and there they form Hessal bodies or Hessal corpuscles. So thymic stroma has three kinds of cells: nurse cells or maintaining cells, endocrine cells or producing cells, and cells of Hessal corpuscles or Hessal bodies. And also thymic stroma contains macrophages. They are present here as well as in other organs of immune system. And also this stroma is filled with numerous leukocytes, and those leukocytes are T lymphocytes because it's thymus, and thymus contains T lymphocytes. It provides development. So here we can see numerous T lymphocytes, and we can see that in cortex they have more close localization. They are located close to each other, and in medulla they are located on the distance from each other. That's why it results in different darkness of cortex and medulla. Medulla is light and cortex is dark because it contains very numerous lymphocytes, and usually they are smaller, and because of this they are located even closer because of small size. And here we can see epithelial stroma of thymus and reticular stroma. At the first view, we can see that both pictures are very similar, but if we remove lymphocytes, we can see that stroma is different. Here we can see epithelial stroma, and in other organs we can see reticular stroma. So stroma, basic cells, supportive cells are different. Here we can see Hassel corpuscle or Hassel body is concentric structure in the thymus, and also its specific feature of thymus. When you see this concentric structure in medulla, you should conclude that it's thymus. It's not spleen, it's not lymphatic node or something else. And here we can see leukocytes, and we can see that cortex is darker and medulla is lighter because of different distribution and different size of lymphocytes. They are located closer to each other in the cortex, which results in the darker staining of the cortex. And also we have capillaries in the thymus, which provide blood supply of thymus. There are capillaries, arterioles, venules, and capillaries in thymus are of continuous type, because we have blood-thymus barrier here. We don't need high permeability of the capillaries. We should protect thymus from damage from cells of blood, and also some immature lymphocytes may excite from thymus and may damage own cells, may kill own cells, and that's why we should provide selection of immature T lymphocytes before this. That's why we need blood-thymus barrier. So how this barrier is composed? We can see here a continuous capillary of continuous type, and it's basement membrane of the thymus, of the epithelial stroma of thymus, and between them we have gap between two basement membranes, and this gap is filled with special macrophage. It's borderline macrophage, which fills this space, and also it provides immune defense. It provides filtration and immune supervision on those cells which are migrating via this barrier, and substances which are migrating via this barrier. Usually this barrier isn't permeable for different cells, and only some substances may pass through this barrier because of complete type of continuous type of the capillary, second basement membrane, and also presence of the barrier macrophage. In medulla, we have another type of blood vessels. We have venules with high endothelium. Those venules aren't present in the cortex. So cortex, it contains immature lymphocytes, and we should form a strict barrier in the cortex. But in the medulla, we haven't this barrier, and blood vessels inside the medulla are permeable for the lymphocytes. So here we can see blood cells, and in the cortex we have immature cells. They undergo their maturation, and when immature cells pass through this barrier and go to the blood, to the tissues and organs, they may kill some own cells, and it will cause autoimmune diseases, and it's very dangerous. To prevent this, we have blood-thymus barrier in the cortex. But medulla, it contains only mature lymphocytes. They cannot damage own cells. They have undergone selection process, and they were checked to the autoaggressive receptors. That's why we haven't this barrier in the medulla. We have venules with high endothelium, and via this endothelium, lymphocytes may pass through this endothelium and go to the peripheral organs of the immune system. So cortex isn't permeable, and blood vessels of cortex they aren't permeable for the cells, and in the medulla we haven't this barrier, and medulla contains mature cells only. So if we have blood-thymus barrier with endothelium, basement membrane, barrier macrophage, and another basement membrane of epithelial stroma of thymus. There are components of blood-thymus barrier. And also you should know that cortex and medulla of thymus, they have separated systems of blood supply. Cortex, it has own blood supply, medulla has own blood supply. That's why blood in different parts of the lobule isn't mixed, and it prevents passing of leukocytes from cortex to medulla via blood vessels, and it keeps complete blood-thymus barrier. So cortex and medulla, they act like two distinct regions in the thymus. And here we can see cortex and medulla. Cortex has blood-thymus barrier, medulla hasn't. And medulla, it provides homing, and also blood vessels of medulla with high endothelium, they are permeable for lymphocytes. And here we can see how lymphocytes are migrating through the wall of venules with high endothelium. Such venules are present also in another organs of immune system, and they provide homing when lymphocytes are coming back to own home, or they are released from home and they are going to the peripheral parts of our body. So here we can see that lymphocyte after the differentiation goes to the medulla, and only from medulla after selection it may pass through the wall of blood vessel and achieve blood. And here we can see migration from cortex to medulla and from medulla to blood. And this blood vessel isn't permeable. And lymphocytes, they have ability for the blood transformation, and after their development, after their contact with antigens in the peripheral organs and systems, they can come back to immune system, for example, to T-dependent zones, and there they undergo some mitotic divisions, and then they migrate into the peripheral organs and tissues. And also very important stage of the development of the life cycle of thymus, it's involution of thymus. Thymus is well developed in the childhood. Here we can see children and adult, and we can compare size of thymus. Till puberty or during the puberty, thymus undergoes age involution. It's normal process, and it's particularly replaced with connective tissue. And thymic cells, T lymphocytes, they migrate into T-dependent zones of the peripheral organs of immune system, to lymph nodes, to spleen. And it's enough to provide immune functions. As thymus undergoes age involution, stroma of thymus is replaced with connective tissue, with adipose tissue, and it becomes even invisible. But also we have another condition which is called accidental involution. It takes place during childhood in some children which have severe diseases, for example, stress, trauma, very severe infection, and it may cause this stress may cause accidental involution. What happens during this involution? T lymphocytes, immature lymphocytes, they migrate into the peripheral organs of immune system to provide immune reaction, and thymus becomes to be empty. And empty stroma of thymus without T lymphocytes, it undergoes a rebuilding, and it's replaced with connective tissue, adipose tissue, and it's called accidental involution. When thymus lost own T lymphocytes, and stroma of thymus undergoes collapsing and replacing with connective tissue and adipose tissue. It's called accidental involution. It's abnormal condition. And each involution which takes place during puberty, it's normal condition. And peripheral immune organs, for example, lymph nodes and spleen, they have T and P dependent zones. So in the lymph node we have cortex, paracortical zone, and medulla. Paracortical zone is T-dependent zone. There are T lymphocytes, and cortex and medulla, they are B-dependent zones. And in the lymph follicles of spleen, we have 4 zones: periarterial, germinal center, mental, and marginal zones. Periarterial zone is completely T-dependent zone. Germinal center is completely B-dependent zone. Mental zone and marginal zones, they are mixed, but B lymphocytes prevail in these zones. And after production of lymphocytes in red bone marrow in thymus, they migrate in own zones, in T and B dependent zones. B lymphocytes, they complete their first stage in the red bone marrow. T lymphocytes, they begin their development in red bone marrow and continue in the thymus, and then they migrate in the peripheral organs. In their zones, they undergo antigen-dependent differentiation, and then they migrate into the tissue where do they provide their functions. And after that, they may migrate in the peripheral organs of immune system, so they turn back from third stage to second. It's called homing. They undergo development, they are resting there, and if it's needed, they again migrate into the place of immune reaction. So students, we have discussed histology of the organs of immune system. That's all, and thank you for your attention.